Hi and welcome to this video on genetic engineering, which is also known as genetic modification, and produces genetically modified or GM organisms. Genetic engineering is a process which involves modifying or changing the genome of an organism by introducing a gene from another organism to give a desired characteristic. Take this plant for example. A grower's income depends on how big they can get this plant and what they can sell it for, but this plant may get eaten by insects, also known as pests, which will decrease the grower's yield, and the grower would have little choice but to increase their use of insecticides, which are strong chemicals that kill pests but can also harm beneficial insects too, and mean that we, the consumer, may be eating food coated in nasty chemicals. Cue a firefly, an insect that can glow in the dark. If we isolate a chromosome from a firefly cell, unravel the DNA of that chromosome to locate the bioluminescence gene that makes a firefly glow in the dark, we can cut that gene out and place it into the plant cell nucleus, where the plant's genome is kept. And if done early in the plant's development, this genetically modified plant cell containing DNA from two different species can divide. The luminescence gene divides with it until we have a complete GM crop plant that glows when bitten. So rather than constantly spraying insecticide to prevent damage, the grower is informed when insecticides are necessary. This therefore limits the use of insecticides to when the pest is in abundance. Overall, the advantage of genetically modified crops is that yield can be increased, and so far genetically modified crops have not caused any problems. However, there are some unknown fears. What will happen if the transplanted gene accidentally makes it into another species? If the gene in question was for herbicide resistance, if this found its way into a weed species, growers will lose a fundamental weapon against weeds. Some genetically modified crops produce poisons that kill insects on their first bite. This is good for the grower, but these insects may be a crucial part of a food chain, and their eradication could affect other species. Some people also blame genetically modified products for a rise in food allergies. Animals can also be genetically modified or genetically engineered. This must be done at the fertilized egg stage if IVF was carried out. Here, cows have been modified to produce low lactose milk, and sheep can make medicines like growth hormone in their milk. Overall, the advantage isn't yield here, but instead it is medicinal. Nutrients can also be mass-produced in areas deprived of that nutrient. A good example is golden rice, which is modified to also produce vitamin A, which helps with eyesight in countries that would otherwise be deprived of this nutrient. In addition, some microbes have been engineered to reduce global warming by taking in excess atmospheric carbon dioxide and producing much safer substances that don't induce the greenhouse effect. This last part of the video is for higher tier only, and it goes into more detail about the process of producing a genetically modified organism. Here I will use the example of producing GM insulin. Here, an insulin-producing cell from a human pancreas is isolated. Thanks to the Human Genome Project, the chromosome required is easily identified and removed. Enzymes then specifically cut out the insulin gene from this chromosome. Meanwhile, a bacterial cell contains a plasmid, which is a small loop of additional genes that can easily be taken up by another bacterial cell. And this has its plasmid removed and cut using the same enzyme. And the human insulin gene is inserted into the gap that this cut has created in the plasmid. This creates a recombinant plasmid. The word recombinant simply means that it contains DNA from different species. This plasmid is accepted by a bacterial cell, and this recombinant cell divides by binary fission to produce clones of the human gene, and this gene is expressed or switched on so that the bacteria starts to produce the human insulin protein en masse. This insulin is then collected and purified and injected by a diabetic patient who no longer produces their own insulin to control their own blood sugar levels after a meal. As diabetes is on the rise, an advantage of genetically modifying bacteria in this way allows more drug to be made to meet the growing demands. Previously, insulin was extracted from the pancreas of pigs, which often led to side effects which genetically modified bacteria has overcome. However, as each protein is made of amino acids, bacteria, being simple creatures, do not have the means to make all human proteins. I also wanted to briefly refer to the plasmid as a vector, a way of transporting the gene from one species to another. Another vector could be to use a virus instead. Here the viral DNA may have extra genes added, and as the virus infects a cell, it often takes its recombinant DNA to the target cell's nucleus, inserting the recombinant DNA into the genome of the organism, and that may allow the organism to have a better chance of survival. So that basically covers genetic engineering. Please have a go at some of the exam style questions that are associated with this video to put to practice what you have just learnt. Do check in again soon.